going to turn to that summary table that you have at the end of the metabolism section, chapter 3, on page 5, and just review it. I asked you guys to complete over the weekend, or over the evening, excuse me. So we'll just compare your answers to, and I sort of summarize everything. To remind you, we may not cover the page 6 material for metabolism. If we don't, it's okay. And then we'll go right into chapter 4. So this is just a simple way to just summarize the highlights, the high points that we covered over cell metabolism, starting with glycolysis. I'm going to skip over to the cell location. Where does glycolysis occur? In the cytosol, you can say cytoplasm, or you can say intracellular food, but not in the what? Not in the mitochondria. Okay. How much ATP will, the net means usable, how much usable ATP is produced following glycolysis? 2 molecules of ATP. And under aerobic or anaerobic conditions? Anaerobic. And I'm assuming, like, what happens next? Well, the big thing is, what's the major byproduct here? Pyruvate. How many? 2 molecules of pyruvic acid. What else was produced? NADH and no FADH2, just NADH and hydrogen ions are the main things that we've been focusing on. That's not the end of the road for glucose catabolism. If what? If oxygen's available, then it's transported into the mitochondria so that another phase of processing can occur. I've used the word what? The term intermediate step. You can call it the linking step, but it's certainly not the Krebs cycle. It's not the Krebs cycle. It happens prior to the Krebs cycle. And so, how much ATP is synthesized during the intermediate step? No ATP is synthesized. Are any electrons utilized, oxygen atoms utilized? No, so this is anaerobic. We said already it occurs where? In the mitochondrial matrix. I cautioned you to go ahead and use the word mitochondrial because matrix is just a generic word. It's even the title of a movie, right? So it occurs in the mitochondrial matrix. And what does it produce? It produces acetyl-CoA. How many? 2 molecules of acetyl-CoA and NADH and hydrogen ion. And we see our first source of carbon dioxide. So we're in the mitochondrial matrix. We've converted pyruvic acid into acetyl-CoA. Then what? Acetyl-CoA can start the Krebs cycle, which also is happening in the mitochondrial matrix. Does it produce any ATP? 2. How much per turn? 1 per turn, but collectively 2. Under aerobic or anaerobic conditions? Anaerobic. And what is it making for us in great amounts? Our electron carriers, which are NADH, how many? 3 per turn. So collectively we see 6 NADH. What else? Well, yes, we mentioned ATP. And FADH2, how many? 2 collectively. Hydrogen ion. Oh, the number, so it would be 6 hydrogen ions, 3 hydrogen ions, and more what? Carbon dioxide is released as well. So at this point, we've only seen 4 molecules of ATP, and we're trying to make ATP. So now, we can use NADH and FADH2 to drive the electron transport chain, which occurs under what type of condition? Definitely aerobic. Where? Yes, across that inner membrane of the mitochondria. And how much ATP can be produced here? Upwards 32, 34, so that range is okay. 32 is okay. What else is produced? Water is liberated or produced. And that's kind of it, right? And we can summarize the ATP synthesis over here so we can get about how many molecules of ATP. Following a complete, somewhere around 36 molecules of ATP produced under ideal conditions. If everything's just right inside of the cell, glucose can produce upwards of 36 molecules of ATP. That's the high end. 32 is significantly greater than 2, or 1 that we saw kind of process along the way. How much oxygen do we make? How much oxygen do we make? 0. A lot of people misspeak and say we make oxygen. We don't make it, we're doing what with it? Using it. We don't make oxygen. Which organs, which systems can make oxygen? What'd you say? Things like plants that can photosynthesize are much better at making oxygen. We use it. Does that make sense? A lot of times people get hung up in all the, or lost or something and misspeak inadvertently and say we make oxygen. We don't make enough oxygen for us to not breathe. We need to breathe to obtain the oxygen, so we don't actually synthesize it. That is a summary of respiration. What questions do you guys have after that review? Talk about it 2 full days, basically. Yes? Anaerobic. The only real aerobic step is which? The electron transport chain. Good. Additional questions? All right, if we have time, we'll come back to the lactic acid pathway. If not, we're okay. So let's go ahead and transition to chapter 4, which is dealing with cell transport mechanisms. We're going to talk across the cell membrane and then within the cell. Just to put it in perspective for you, a cell, like I tried to mention before, is not isolated from the environment. It must exchange things like oxygen to tie into what we just studied, or carbon dioxide out, or nutrient glucose into the cell or whatever. But not all molecules have the same chemical property, and therefore they can't cross a cell membrane equivalently. Okay? Because we know about the features of the membrane. We know the membrane has a huge phospholipid bilayer, which is going to limit particles that are charged or big. Okay? But we know that we need charged and big particles to enter and exit the cell. So we've got to have the ability to get not only just tiny little non-polar particles, but other molecules across the membrane. So the cell can maintain what? Homeostasis. Okay? So if we use some terms, we talk about permeable or impermeable. Permeable means the membrane is permeable to something, that something can traverse the cell membrane, or the membrane may be impermeable to something, where that particle cannot cross the cell membrane. But we generally, if we're talking about the membrane as a whole, it's described as being selectively permeable. So it's permeable how? To certain but not all. Okay, so it has some level of regulation of what crosses its cell membrane. Selectively permeable. I think you've probably used that term before. In this concept we've already utilized, your book uses the term forces, and I tend to use the word gradients. But there are forces which allow for a particle to move across a membrane. The force is what is going to move the particle, and the particle is something, it's just a generic word, atom, ion, some molecule, whatever. So it's just a generic word without being specific, like sodium ion or glucose, whatever the case may be, just whatever. We're looking in general, how something can move across a membrane. Today we're going to talk about chemical forces or chemical gradients. I have an asterisk by electrical forces because we will go into that in better detail, in great detail, starting on Tuesday when we look at the nervous system. So we'll just talk about chemical concentration at this point, and we'll add in charge next week. But we will talk about the electrochemical gradients in the nervous system coming up. What we're going to look at is passive versus active mechanisms, which we've become familiar with. Passive transport moves a particle down a gradient, so that means from greater to lesser for that particle. So for example, if I want to move glucose into a cell passively, more of it has to be where? In the where? Where would it be? So anatomically, where would it be in the greatest concentration? If I want to move it into the cell, it must be in greater concentration outside of the cell. Okay? The diparticle that we added to your cells the other day, it was obviously in greater concentration outside of the cell when you dropped it on the outside. But we saw diffuse in some of your cells down its concentration gradient. We did not illustrate active membrane transport, but we became fundamentally familiar with it, and where the gradient is reversed. And so in order to reverse the gradient, a particle needs to be, it takes energy. You need to move something considered, the term you can use is uphill or against the force. So if I want to push something uphill or move a particle from lesser to greater concentration, it's going to require an input of energy. The most common form of energy is which molecule? ATP. Is it the only thing that can energize? We gave a great example yesterday. What could energize a protein to actively transport? Electrons powered the hydrogen pump across the intermitochondrial membrane. So at this point, we can understand passive transport moves a particle down without any input of energy. Active moves up with energy. It also requires a what? A carrier or a transporter. And it's that transporter that uses energy. Some examples are the categories of active versus passive transport. In passive transport, there's simple and facilitated diffusion. And if you have your handout on the back page, those are the two that we illustrated. We illustrated simple and facilitated diffusion. We did not have osmosis in the handout, but we did the hands-on experiment with osmosis. So we have something visceral to kind of go back to. I like to include in there, and this isn't technically, I don't know if it's right or not, but I like to include filtration in our list. And it's not so much a chemical gradient, but a pressure gradient, where something would move from which pressure to which? Higher pressure to lower pressure. And so we'll see this in our capillaries and in our kidneys, where filtration is important. Conversely, our active transport mechanisms, there's primary active and secondary active transport, which we'll illustrate in a moment. And then I want to include our forms of vesicular transport, or transporting what? Vesicles. like for example from our Ruffyard to our Golgi. That requires energy. We'll conclude with those. So let's look at some of these in better detail. And again, this will be a review for you from a summary from our lab on Monday and Tuesday. In simple diffusion, a particle moves down a concentration gradient, and the key word, it is random. The particles don't know to go across the membrane. They don't know anything. They're just colliding, and they're trying to reach what? Equilibrium. Or is described here, a steady state. So the particles are just trying to reach equilibrium between the extracellular fluid and the intracellular fluid. So just simple diffusion. If we look at simple diffusion, let's add some detail in the margins of your notes. What type of particles do you think could simply diffuse across a membrane? Water can. It's one of the weird exceptions. That's not a great example. It can, though. You're right. What else could simply diffuse? What chemical properties about a molecule could simply diffuse? Or give us another example. Oxygen and salts actually cannot simply diffuse because they're what? Charged. So charge-wise, what type of particles are more likely to simply diffuse? Nonpolar. So we're going to add in our notes, nonpolar. In what? Probably particles that are small. I think generally about less than 100 Daltons is kind of the textbook value about what could simply diffuse. Something less than about 100 Daltons. So small and nonpolar particles are more likely to simply diffuse across the cell membrane. The next image that I have just gives you a visual, like we saw with our simple diffusion experiment with the dyes in the agar. We saw the particles scattered. If we had to let it run overnight, the bulk of, if not all of that agar would have been colored blue or yellow. So, as illustrated, our respiratory gases, oxygen and carbon dioxide, can simply diffuse, which is good. We don't have any limiting ability to maybe move oxygen in. If it's available, it's going in. Also, if we look at our kidneys, I mentioned, have you ever heard of urea? It's a waste product, but it's small. It can simply diffuse across our, between our tubes and our capillaries and our kidneys. So we'll address that at the very end of the semester. If we look at diffusion, we have 5 things that influence the rate of diffusion, and as I mentioned in lab, I said really we can add 6. And the 6th thing that's implied in there is temperature. So let's just summarize what we learned in lab this week about rate of diffusion. So we studied items number 1, 4, and 6, right? 1, 4, and 6 are the things that we studied in lab. So 2, 3, and 5 we did not quantitate, but we actually discussed. So looking at item 1, if the concentration gradient, what's the relationship between concentration gradient and rate of diffusion? As she said, as one increases, so does the other, and so that they are directly proportionate. Very good. So if the concentration increases, so does the rate of diffusion. Second item on the list is the permeability of the plasma membrane. What kind of relationship do you think is there? About the permeability of a membrane to a particular substance. Very good. The more permeable the plasma membrane is, the more likely it's going to simply diffuse. direct. The permeability increases, so does the rate of diffusion. The third item, SA, stands for what? Surface area of the membrane where diffusion is occurring. What do you think the correlation is between surface area and rate of diffusion? Very good. Same thing, proportionate. As surface area increases, the rate of diffusion increases. So it's direct. Item 4 we studied. What does the MW stand for? The molecular weight, which is translated into the mass of the particle. What was the correlation between molecular weight and rate of diffusion? Very good. You guys said the higher the mass, the slower the rate. So the relationship is inverse. Great. Big particles just cannot fit through those spaces as easily as small particles can. The fifth item is the distance of the membrane. We didn't acknowledge this. However, not all phospholipid bilayers have the same thickness. Some fatty acid tails may have, say, 16 carbons. Some may have 17 or 18 carbons. As we add carbons to the tail, what happens to the length of it? It's a little bit longer. And so what do you think the relationship is between distance and rate of diffusion? Very good. We said inverse, or in other words, the greater the distance, the rate of diffusion will decrease. The first 5 are part of what's called Fick's law, and then 6 is implied when we think about these basic chemical interactions, but we can't not consider temperature. So what was the relationship between temperature and rate of diffusion? Direct. Okay? If our temperature increases, particles will increase in their motility, so diffusion is enhanced or favored. So that's another direct relationship. What questions do you have over simple diffusion, what it is, or things that might change the rate of diffusion across the cell membrane?
The molar rate of a substance is inverse.
Yes, the molecular weight is inverse. Molarity is concentration. Correct. Molecular weight in mass is inverse. You okay with that? Additional questions? Okay. All right. So another type of diffusion is called what? Facilitated diffusion. Facilitated diffusion is still diffusion, so which way are particles moving? From a greater to a lesser. So down their concentration gradient. But what? What makes it not just diffusion, but facilitated diffusion? There's a carrier, a facilitator, a helper, right? The helper is there to do what? Carry it, right? It gets in the membrane. What kind of particles do you think might need help? Larger and or polar or fully charged. So what we're going to add, we've got it. charged, polar, and add in there larger as well, large particles. So in facilitated diffusion, particles can still move down their gradient, but because of the chemical nature of the membrane, they need those fatty acid, that hydrophobic region, pushed out of the way. So it may be a channel, it may be a carrier, but the whole point is it carries a specific molecule from a greater to a lesser concentration for that molecule, and does not require what? Does not require energy. If we look at a snapshot, it almost looks like active transport, but we can see that it's not because of the gradient, right, the directionality at which the particles would move. The rate of facilitated diffusion is going to have some same type of characteristics, so like temperature is going to play in there, presence of the molecules, but because we're adding a protein carrier, we can add a little bit of additional detail in there. So a big one that we're most familiar with is the concentration gradient. So clearly that's going to dictate how fast the particle will diffuse down this gradient. If there's a bigger gradient, faster, we can handle item 3. Items 1 and 2 might not have thought about. There may be a huge concentration gradient for a particle, but if there aren't enough carriers, it's going to be limited at the rate at which the particles could diffuse. And then so, item 2 is just the direct relationship. If there are plenty of carriers, the rate of diffusion can, facilitated diffusion can occur maximally. If there are few or no carriers, even though there's a gradient, would there be any diffusion? No, or minimal. And then you also have to take into consideration is how efficiently the carrier works. Is it fast or is it slow? So that's going to factor in there as well. The major facilitator that we'll study this semester is the glucose carrier. You remember in chapter 3 we talked about glucose is catabolized in the cytoplasm? Well, it's a big particle, so it has to get facilitated into the cell membrane, across the cell membrane, so it can be utilized. And also in the renal system, you know that you have glucose in your sugar, or glucose sugar in your blood. It actually gets filtered at your kidneys, but it should be reabsorbed and taken. So we have plenty of what at our kidney tubules? If I want to take glucose, move it from one spot to the other, carriers. So I just have glucose carriers. Lots of glucose carriers at my kidney tubules. Put it back into the blood. What questions do you have about facilitated diffusion or contrasting it to simple diffusion? Both forms of what type of transport? Passive transport. A third type of passive transport is called osmosis, which we've already studied. And so I'll go through fairly quickly, but you still may have questions at this point. So if you have questions, that's why I'm here, so ask them. Okay? And the key thing with osmosis in the body, in a biological system, is it's still passive transport. It may be simple diffusion, but usually it's facilitated diffusion because water is what? It's charged. It's not too big, but its charge is polar. It's like the classic polar particle. And so all logic tells us that it shouldn't simply diffuse, but and because it's so little. So most of our osmosis occurs across cell membranes because our cells have aquaporins. I think I've got that coming up for you in your handout. So I'll address that in just a second. But it may be simple, it may be facilitated. But the point is, water is moving where? Down its own concentration gradient. From an area where there is more water to an area where there is less water. We're going to put the cart ahead of the horse a little bit. If I want to apply the terms that we used on Monday and Tuesday. From which solution would water flow from and to? Which tonic solution would water flow from? Always from the hypotonic to the hypertonic. hypotonic has greater amount of water. And so the key is, of course, is to find across a selectively permeable membrane, and it's going to happen in greater proportion when the solute concentration or the tonicity is different between the extracellular fluid and the intracellular fluid. We'll talk about it across the cell when we use these tonicity terms, extracellular fluid, intracellular fluid, but really I can apply hypertonic, hypotonic to anything, any two fluids. I could compare the drinks up the front here, like the water versus the yummy frappuccino looking thing, right? Mm-hmm. Which one would be more hypotonic? The water. The one that's hypertonic would be the yummy drink, right? So even though those aren't, I could use the same terms. I could say the bottle has a hypotonic environment and the Starbucks cup has a hypertonic. But, so I'm just cautioning, you could use those words anywhere, but we are applying them to the extracellular fluid and intracellular fluid. Does that make some sense? And the concentrations are not the same. So I mentioned a moment ago, the whole point of osmosis is try to equilibrate water and solute concentration. And I also mentioned that the cells may have these things called aquaporins. May seem like a big word, but if you work through it backwards, it's a pore that's permeable for water. So basically it's a water channel. So it allows for facilitated diffusion of water more easily. What questions do you guys have at this point about how to define osmosis? As we mentioned, it happens to help equilibrate, and we learned from our experiment that when osmosis occurs more in one direction than the other, we actually change the fluid content of the cell, which changes shape, which changes its function, right? Well, it gets broken. And so if we change the fluid content of the cell, we really mess up the concentration of solutes and the shape of the cell. So it could be catastrophic to a cell, even if it's not necessarily if it's just losing water, but even gaining water can cause damage to the cell. And the whole point of my abbreviation is to try to equilibrate out the solute. Anybody know what my brackets are representing? Concentration. Okay, so I'm just trying to abbreviate the solute concentration. The whole point is to try. Whether or not it happens depends on the magnitude of the condition. But the whole point is equilibrate solute and water or solvent concentration. We became familiar with some terms to help better understand this. We talked about the tonicity. And the tonicity influences the cell shape. And by changing the internal water what? Volume. And my abbreviation stands there for volume. We learned that not all solutions have the same tonicity or the same amount of solutes in them. If they did, we would have nothing but water and no other, right, nothing. We wouldn't be exciting at all. And so some solutions are equivalent in composition, and we call those what? Isotonic. Now I want to caution you again, I think you guys have a good grasp of the prefix, hypo, iso, hyper, but the suffix, the tonic, deals with what? The solute. More specifically, the solute. Not the what? Not the solvent, not the water. So we read through it, and we sometimes, if we don't really get a good command of it, we get backwards. We just see the hyper, and then we presume water, and we get all kinds of backwards. So iso means same. Tonic means solute concentration. So if two solutions are isotonic, solute concentration is the same. We imply then the water concentration is the same. Is there any net movement of water? There is no net movement of water. Does that mean there is no movement of water at all? No. Water's still moving. Particles are still moving, but it's what? The same. Particles move in, and then particles will move out at the same rate. So the cell maintains its shape, maintains its volume, maintains its function. Very good. Conversely, we know that we can fall out of conditions, ideal conditions, and solutions may become more hypertonic or hypotonic to one another. Hyper means what? More. Tonic means solute. So those solutions that have greater solute concentrations, if that's on the outside of the cell, like we did, we put our cells in a hypertonic solution. If the outside of the cell is hypertonic, what impact does it have on a cell? It shrinks because water is moving out more than it's moving in. Changes the structure, changes the function. Right? It'll do that. Someone used the word dehydrate. Basically, hypertonic solutions dehydrate cells. It causes them to lose their water. Conversely, solutions may be hypotonic. Hypo means less. Tonic means solute. So we can extrapolate that to mean it has a whole bunch of water. And so if a cell is surrounded in a hypotonic environment, they'll swell up. Very good. And so a few years ago, do you remember became nationally, publicized, or addressed is a better term. Remember when someone did the water challenge? They just drank so much water. You think, well, water's not harmful, right? It is if you do what? Take in too much. Okay? So in that case, the person made their environment, their body really hypotonic. Cells began to take on too much water. They maybe even began to swell and maybe bust. But probably more immediately, what we're going to learn in the second unit is we diluted out the concentration of our solutes, like sodium, potassium. You remember at the beginning of class, I said we basically call this sodium, potassium, calcium class. We mess up the function of our nerves. We mess up the function of our heart. Okay? And so these hypotonic environments, if they're not, in an extreme condition, it can be just as lethal as a hypertonic environment. So a common question is, like, if I go to the hospital and they put an IV into my body, and so I've been complaining of migraine headaches, so I go to the hospital and they put an IV into me. Okay? What type of solution should I want? Do I want just water in there? Isotonic. Normal blood plasma. If it's isotonic, the fluid movement should be ideal, acceptable. Just have a few more images to illustrate this. Under animal tissues, isotonic is preferred. This is not the same in all organisms, but we're studying the human body, so we prefer an isotonic solution. And as we learned from our experiment, we definitely saw the cells shrinking and losing the dye particles. As well as in our hypotonic, we saw the cells taking on water, but we didn't let it run enough to actually bust the cells. But we could have maybe watched them to see if it would happen. Some real images, some scanning images of a cell, red blood cell in an isotonic environment. Let me zoom that back up. You can see the concave nature. That's normal. We'll talk about in chapter 15 why that is. And then this cell has been exposed to a hypertonic solution where the fluid has moved out and it's just completely messed up the shape of that. You can see how that would be bad trying to get through your blood, right? You all hung up and jammed and stopped blood flow. As well as a hypotonic environment. That too could get stuck in the circulatory system of your blood vessels. So we prefer our cells to be what's considered isotonic. We don't want to have these extremes. Any questions at this point about osmosis, or did the lab help a little bit? Yeah. The next item I want to talk about is called filtration. As I mentioned, it's not necessarily kind of one of the technical categories that you have in passive transport. I'd like to address it because we're going to use filtration a little bit this semester. And we're still moving down, but it's not a concentration gradient, it's a pressure gradient. The particles can be moved from an area of greater pressure to an area of lesser pressure. So for example, if I want to breathe in, air would move down the pressure gradient. Or breathing out is a better example. Air would move from a greater pressure in your lungs out into the atmosphere. For your capillaries, blood plasma is forced down this pressure gradient in your interstitial fluid. And then reverses the pressure can move particles from a greater to a lesser area. The deal is with the pressure gradient, it's not specific. Up to now we've been looking at specifically, say, glucose, or specifically water, or specifically sodium, whatever. Anything exposed to the gradient can be pushed down down the pressure gradient. So again, we'll mention this in our capillaries and how our urine is formed as well. Everybody's thumbs up so far? Okay. All right, those are different types of passive transport. We're now going to talk about active transport. Active transport, we're going to define it with our textbook definition of ATP, but we now know because of the mitochondria, there are exceptions to the rule. But ATP is the primary energetic source for moving particles. And we're going to look at active membrane transport and then include in there vesicular transport, which your book uses the word via compartment. I don't know why, but they do. But movement of vesicles. Active membrane transport and vesicle transport. In active membrane transport, it's going to be kind of familiar in that there's a carrier necessary, but the thing is the carrier is going to need energy. The carrier is going to carry a specific molecule. So you don't have just a generic active transport that will carry anything at once, like glucose or sodium or calcium or whatever. It's specific. If a cell wants to regulate sodium, it's going to have a sodium carrier. If it wants to regulate calcium, it's going to have a calcium transporter, or we call them pumps. And so it's really specific, but it's also reversible. So it binds it and does what? It binds the particle and then attaches it and then moves it and releases it. Okay? So it's got to be reversible. So it attaches it, moves it using energy, and then lets it go. If we look at active membrane transport, we can really subcategorize this. You're probably most familiar with primary active transport, which I have abbreviated with the 1 and the degree symbol. That's primary active transport. And in that case, that was also from your handout that you turned in on Tuesday, the third little image that you acknowledged. And in that case, the carrier directly uses ATP. In other words, the carrier directly gets phosphorylated. Gets energy and directly pumps a specific molecule across the membrane. There's primary active transport that directly uses ATP, and secondary active transport that, how do you think uses ATP? Indirectly. So I'm going to come back to that in a moment because some of people have a little hard time understanding that, so I'll better explain that. But at this point, we're going to say primary directly uses ATP and secondarily uses it indirectly. Let's first look in a little better detail at primary active transport. If we were to set it up in a simplistic fashion, we can see a membrane. Now this could be the cell membrane facing the extracellular fluid or the intracellular fluid. It could be a carrier on your smooth ER or the mitochondria, whatever. But we can certainly see an integral membrane protein. You can see it carries a particle. We can kind of see almost like a channel, but it's regulated. We can definitely see that it can be phosphorylated. And what dictates the directionality is, we don't have any quantification, but it says high, low. So in this case, active transport would move the molecule we've been using against its gradient. To push against that gradient, we need some form of energy to cause that protein to change shape and physically move the molecule across the membrane. So what it does is binds a particular molecule in its relaxed state, and then it gets energized and pumps the molecule against its concentration gradient. And then once it's out there, do whatever it's supposed to do. So as we identified, in primary active transport, the energy does come directly from ATP. So it's directly receiving the energetic source. Identified as well, the transporter, if we're using ATP, is phosphorylated, which we know changes its function, gives it what? What is transferred into it? Energy. The phosphate's transferred to it, transferring into it energy. Do you remember that terminal phosphate of ATP has all that electrostatic repulsion? And so we're transferring that energy into the protein. And so a lot of these things are commonly called pumps. If you use the word pump, you're implying what type of transport? Active transport. So be really careful when you use the word pump. Don't just use it willy-nilly, at least in this class. Okay? Because pump, if I read the word pump, I'm reading what? Active. In which way in respect to the gradient? Against. And I'm not trying to be nitpicky, but we understand at least if we change the direction of something, we're going to completely mess up the function of it. So I'm not being nitpicky. I'm trying to illustrate to you how our body works. Okay? And so the word pump implies active transport, moving a molecule against its gradient. The major pumps that we're going to use, the primary pumps that we're going to talk about this semester, include the sodium-potassium pump and the calcium pump. Okay, so from here on out, nervous system, muscle system, you will be really familiar with these things. You'll notice that they are named for which they pump. The sodium-potassium must move each sodium and potassium against their own concentration gradient. What we're going to learn is the directionality is not the same. Calcium pump pumps calcium. We'll talk about the calcium pump mostly at the smooth ER. Do you remember we said the smooth ER is specialized for calcium storage? It's got to get in there. You can pump it in there. I'm going to go through the pump because from here on out, starting on Tuesday, probably on Wednesday, maybe in our lab Monday, we'll become, I think, familiar on Monday with the sodium-potassium pump in the nervous system. So I'd like to go ahead and address how it works so that Monday's lab will go a little bit smoother for you. The sodium-potassium pump, it utilizes ATP, or you could properly say hydrolyzes ATP. The sodium-potassium pump is phosphorylated, which we know means it gets phosphate and energized. What it does is it pumps each sodium and potassium ion, but in a specific direction. The sodium-potassium pump will pump 3 sodium ions out to the what? Extracellular fluid, and pump 2 potassium ions to the intracellular fluid. Based on this directionality alone, I know now that there's an ion imbalance wherever there's a pump. Where would sodium be in greatest concentration in a cell that has a sodium-potassium pump? 50-50 in or out? Out. How do you know? The pump is pumping it out. We know it's active transport against the gradient. Conversely, potassium must be at greatest concentration inside of the cell, which we know because the pump is pumping in, actively so it's concentrating potassium inside, concentrating sodium where? Outside. And notice that disproportionate, it's 3 to 2. We'll come back to that on Monday and Tuesday in our lab. So I'm taking 3 positive ions out and returning only 2 positive ions in. What's beginning to happen in cells where we find a sodium-potassium pump? We're going to change the polarity. Think about it in your bank. I take $3 out, put $2 $2 in, $3 out, $2 in, your bank account's gonna plummet. And so we're gonna plummet the charge across some cells. The whole point is to create concentration gradients. We've seen this in play. Where did we create a concentration gradient in discussion yesterday? The electron transport chain. Why do we create gradients? So we can use it. So the sodium-potassium pump is going to be in tissues to help establish concentration gradients, which thusly will regulate our cell volume, because our ions influence movement of what? They could be salts. Okay, where salts are, influence is where what is? Huh? Fluid, like water. Okay, we just talked about an osmosis. If there's a difference in solute concentration, we can change where water moves. So the pump not only establishes gradients so we can use them, it's going to fundamentally make sure the cell has the right volume of fluids in it, maintaining its equilibrium is shape, right? Fluid shape. Okay, so this has influence osmotic effects. Those are some primary roles of the sodium-potassium pump. Figure, I don't know if this is your image or one like, I think this is from your textbook, figure 4.4. We can see an oversimplified view of the sodium-potassium pump. We can see where it binds 3 sodium, picking it up from the intracellular fluid. It will not change until it's what? Phosphorylated. Gives it energy. And notice how when it gets phosphorylated, it changes the fluid which it's exposed to, and no longer prefers to bind to sodium. So let's go with sodium. And then it can now favor the binding of what? Potassium. In order for it to go back to its resting state, though, the pump needs to be what? Dephosphorylated. Where it can unload potassium and now reload sodium. So just pump continuously. I think I've got several images. Oh, I got an animation. In the blue, we have the pump. Okay, that's just a cell membrane wouldn't be this simple. But we're just trying to isolate the pump only. And it would obviously work faster than this. The sodium is in the purple, and the potassium in the blue. Phosphorylate the protein, transport the sodium, binds potassium, pump it in. If you look at better detail, you'll notice the potassium is in greater concentration where? Where do we see more purple dots? It's changing on us all the time, isn't it? Well, that'd be good, right? So you can see more on the outside, right? Now, it's trying to concentrate the sodium outside of the cell, and then concentrate potassium inside of the cell. That's the sodium-potassium pump. We could have done the same thing with calcium. Calcium would bind, the protein would be phosphorylated, and calcium would be pumped into wherever it needs to be located. Can you see how this protein is directly phosphorylated? And now it's moving particles against their gradient. What questions do you have about primary active transport or the sodium-potassium pump? At this point. Fairly comfortable? Alright, so there's primary active transport, and there is also secondary active transport. This is an indirect way to move a particle against its concentration gradient. In other words, a particle can be moved against its gradient, but its carrier itself does not directly need to be phosphorylated. However, something had to have been actively transported first. That active transport, something primarily active, creates a gradient other molecule kind of hitches a ride, if you will. An example which I've used, like not a biological example, but it's like a scenario that you could probably appreciate and wish for, is are you familiar with the lottery? The Oklahoma lottery? Anybody want to win it? To win it, what do you need to do? Play. So you need to pony up $1, right? So only those people who directly go to 7-Eleven and put down their dollar and buy a lottery ticket have a chance to win. Kind of. Right? So let's say I go to 7-Eleven, I have $1 in my pocket, I buy a lottery ticket, and I win. How did I win? Directly or indirectly? I directly won. Okay? But won't my children get the payoff? I'm a nice person, I'll probably share with them, right? My boys, can I probably buy them some more Legos or some other crap they don't need, right? So kind of didn't they win the lottery? But did they earn the money and go to 7-Eleven and win the lottery? No, but they didn't directly win it, they indirectly won it. Molecules can indirectly be actively transported, but some other molecule had to be actively transported. Had to go to 7-Eleven and buy the lottery ticket, right? And so the work that was put in by another molecule gives a gradient or a drive for another molecule to be moved against its own concentration gradient. And that's referred to as secondary active transport. We won't really put this in our practice. We'll mention it at the end of the semester, how particles are secondarily active transported out into diffusion. But it's just another form of active transport. It doesn't directly use energy, but it indirectly does utilize it. This small image that I have on the handout on page 3 helps illustrate this. Hydrogen ions, we can see being actively, directly, primarily transported against its gradient. And then substance S. Can you see how it's moving against its gradient? There's a carrier moving against its gradient, but it's only moving because a hydrogen ion is moving down. Right? It had its own gradient, it's moving down its concentration gradient. So had there not been a hydrogen ion gradient, would solute S diffuse, or excuse me, actively be transported? No. So first, primary active transport drives secondary. It's very label for you. I haven't put this in there. What questions do you have about primary active transport or secondary active transport? I want to transition now to vesicular transport, which is the movement of vesicles. And vesicles, up till now we said they move, but it does take energy to move, to form the membrane, and then move the little vesicle from one spot to another. And we can really subcategorize it into 4 types of vesicular transport. The first is called exocytosis. This is a word that we used when we talked about the Golgi apparatus and the formation of those extracellular type proteins, where something was originally in the intracellular fluid, wrapped in a vesicle, and then purged out to the extracellular fluid. Instead of saying purged out, I could say exocytosis. So it's a way of removing substances out of a cell. Conversely, we can also endocytose particles and wrap them in a vesicle. So something that was originally out in the extracellular fluid gets enclosed in a vesicle, and now it's compartmentalized in a particular cell. Once a vesicle is within a cell, it can be transcytosed. Where we'll see this, transcytosis is at our capillary. Endocytosed, moved across the cell, and then actually exocytosed. So it's like physically enter, move, and release. So think about your blood plasma and the capillary cells I had you guys draw a couple weeks ago. Big proteins can move from the plasma across across a capillary cell in a vesicle and then out into what? Which fluid? What part of the extracellular fluid? From the plasma across the capillary cell and into the interstitial fluid. So we'll use this word in chapters 14, somewhere around there. And then what we became familiar with this week is trafficking. Vesicular trafficking, where a vesicle is just transported, say, from the rough ER to the Golgi, or maybe back, right? Or from the Golgi to form a lysosome or whatever. Or maybe when we look at the formation of our vesicles and our nerves to move our neurotransmitter from the cell body down the axon to the terminal, right? That's just called trafficking. That's just physically move, like cargo from one spot to the other. But if we were to look, the energy comes primarily from ATP, but there's another molecule called guanine triphosphate, or GTP, can also power some of the motors that move our vesicles. Exocytosis doesn't randomly happen. It's got to be initiated and regulated. So maybe we use the word ligand and receptor already this semester. So some type of extracellular messenger can trigger a cell to exocytose its contents. So without getting into the technicalities in this course, basically the vesicle is physically carried over to the cell membrane, and because of the way that the lipids interact, the membrane of the vesicle actually fuses with the cell membrane, and so now it just incorporates. What was originally incorporated inside the vesicle is now exposed to where? Where is it exposed to? The outside, to the extracellular fluid. Pictures, I think, will help a little bit more. And if you look at the first image on your right-hand side of your handouts, here we have a vesicle, little yellow things, right, proteins, and maybe they just came from the rough ER and the Golgi, right? That vesicle can be transported over to the membrane of the cell, and notice how the vesicle membrane joins the cell membrane. And at that point, the phospholipids will kind of interact. And so now the vesicle actually becomes incorporated where? In the cell membrane. And what was originally just isolated is now dumped out extracellularly. So this is called exocytosis. And we'll use this a whole heck of a lot in the second unit, looking at our neurotransmitters. We could reverse this process by what? Endocytosis. Endocytosis has a few subcategories, so I'd like to address those. Oops. Let me look at this. Someone was talking about, I think a day or two ago, that cells are just kind of moving stuff out. This is a great image. Can you see the vesicle? And it's just spewing fragments contents out. So I had a genetics class and or an immunology class, and my instructor called it's like forever in my brain, cell vomiting, right? Exocytosis, basically, purging all its contents out. Endocytosis, as I alluded to before, there are some subcategories, 3 subcategories that we'll study, including pinocytosis, receptor-mediated endocytosis, and phago or phagocytosis, however you want to enunciate that. And so this whole point is we're taking in substances. If something's taken in, it may fuse with a lysosome. Why? To break it down. To catabolize whatever that is, like a bacteria or whatever the case may be. So we're going to look at each of these 3 things. The common thing is we're taking substances where? Inside of the cell, wrapping them in a vesicle. Pinocytosis, we'll start with phagocytosis. I say phago, someone else may say phago, whatever, the same thing. And phagocytosis is large scale, so physically something huge is taken into a cell, like an entire other cell is taken into a particular cell. In order to do so, the cell membrane will extend to make these things called pseudopods. Pseudo means false, pod, foot, feet. Has these things, cytoplasmic extensions that look like feet or arms, but they're not. They're just cytoskeletal proteins pushing out the membrane. And they encompass, or like bear hug, something. And when they do that, the membrane's fused, and what was originally on the outside of the cell is now encompassed in this vesicle. And by technical definition, that vesicle that has just been formed by phagocytosis, you can call it phagosome. I don't usually use that word, but that's the proper term. And so these cells that have the ability to do phagocytosis move by what's called a amoeboid motion, so it looks like they're walking, they're pushing the cytoplasm. Be more familiar with like your white blood cells? This is what they can do. When they resolve, like big dust particles in your lungs. And your neighbor keeps sneezing on you and they're putting like their little bacterial pathogens in the air, you're okay, you got macrophages in there. Phagocytosis, like that, making it not harmful. Not picking it, oh yeah, Alright, so we can see an example here. The green thing, identified as a bacterial cell, will be phagocytosed. You can kind of see the little pseudopods projecting out. Engulfs the membrane, pinches. It's very much regulated. It's not random. Very high tech. What happens? But then, what was extracellular is now compartmentalized. What happens to it depends on what the cell wants to do with it. Destroy it or whatever. Do it. Yeah, catalyze it, or if it's a nutrient, right? We may use catalyze it or whatever, but a bacterial cell, you would certainly metabolize it. So phagocytosis is large cell engulfing, or large particles, big. Here's an example. The little white, blue-colored cell is actually taking in the big green thing, so you can see how large scale this is. It's going to begin to pinch off and just break down that thing piece by piece. Take it in, and purge out its content. Kenocytosis. Keno is referred to drinking, so it's endocytosis, but it's like small, like on the opposite end of the size spectrum. Tiny little endocytosis. It's called cell drinking or fluid phase endocytosis. And it's a way in which the cell can sample its environment. And you just go in and just like pinch all their tiny microscopic little vesicles that are formed. And then it'd be nice if this had a lot of them, like tiny little vesicles all across the medium. So penocytosis is still endocytosis, but what? Small, and probably more numerous as well. And another type of endocytosis is called receptor-mediated endocytosis. So this happens when what? What do you think might initiate receptor-mediated endocytosis? Some can do that. Some viruses can act this way. What has to be activated? A receptor. What binds to a receptor? Just generically, we call those things. It may be, I don't know, just generically, anything that binds to a receptor, we said we can call what? Just a moment ago, ligand. So in other words, in receptor-mediated endocytosis, it happens only when what is present? A ligand binds to a specific receptor, and that triggers the endocytotic pathway. So we can look at the image in your handout. You can see a cell membrane. You can see a little indentation. But we notice that the membrane is not so plain. There's some receptors here. What color are the receptors? The purple things. The green things regulate the process. We don't need to go there in this level, but we need to acknowledge when the ligand is colored what in this image? Red is absent. Endocytosis happening there? No. Only when the ligand is present will it initiate the infolding of the membrane and the formation of the vessel. The whole point is it's taking products which way into the cell. I'm going to conclude with this published image, a really great transmission image. You can see the receptors at the cell membrane, the ligands beginning to bind. See the background, forming the vesicle. You see how the membrane associates and pinches and forms a freestanding vesicle. We're out of time, but we made it. So you will be accountable for all which we've covered, and you'll have an exam on Monday. So bring your brain power and a pencil, and we'll have a test on Monday. See you guys in the lab or on Monday.